Okay, we are working our way through viral multiplication. First step was adsorption. Second step was penetration, needs to get in. Penetration, uncoating occurs, and that lets that nucleic acid be released into the cytoplasm. Then we talked about synthesis, and that can happen in a variety of different ways depending on what type of nucleic acid came in. And now we need assembly. So it's replicated all this nucleic acid, lots of strands of whatever nucleic acid it needed to replicate. It has all these proteins. Some have been used as enzymes. Some are structural proteins, so those capsomeres that makes the capsids. What happens next? The next phase is assembly. We need to put all those together to make a fully packaged virion. All right, so let's look at that. All right, assembly. If it is a DNA virus, assembly occurs in the nucleus. So those proteins got made in the ribosomes, but they get transported into the nucleus, and that is where assembly occurs. Seems counterintuitive, but they had to do the experiments and figure it out and marked things, radioactively labeled things, and they figured out that this is the way it happens. So DNA virus assembly occurs in the nucleus. RNA viruses, they're already in the cytoplasm. The proteins are there. Assembly occurs in the cytoplasm. We also, due to experiments, know that the capsid gets formed first. So these capsomeres come together, and we don't know how. We keep saying spontaneously, so that we don't, which there's probably more into it, we just don't know yet. But the capsid, at some point during this process, these capsomeres spontaneously come together and form that capsid. What shape is that capsid going to be? Well, whatever shape it came in is what it's going to be in form. So that happens, and then the nucleic acids get inserted into the capsid. Okay, so that is assembly. During that process, if it was an enveloped virus, some of those proteins are going to be those glycoprotein spikes. So at this point, those proteins that were made get inserted into the host cell membrane so they can be picked up as during release. So they can be picked up on the when the virus leaves the cell and gains its envelope as it leaves. All right, so that's assembly. The last stage is called release. So let's see what that looks like. So if it is an enveloped virus, it has to pick up those glycoprotein spikes, which have been inserted into the host cell membrane. So this process of gaining its envelope is called budding. So if it is an envelope virus, it must leave by budding. So what happens is we had that capsid form during assembly. The nucleic acid got inserted into the capsid. So now we have a fully formed nucleocapsid, capsid plus nucleic acid. It then moves to the cell membrane. The cell membrane had already been lined with viral matrix proteins. So during that synthesis stage, some of the proteins that made were called matrix proteins, and they lined the cell membrane of this host cell. Other proteins were those spikes, those glycoproteins that are called spikes, and they have already been inserted into that. Nucleocapsid binds to these viral matrix proteins. That starts a chemical reaction that kind of induces a kind of an exocytosis, a modified exocytosis that we call budding. The nucleocapsid moves in, this kind of pinches off this phospholipid bilayer, this membrane portion, and becomes a fully infectious virion that is released into outside of the cell, into the host. This is called budding, and it's how enveloped viruses always leave the cell. Now, it mostly happens on the cell membrane, but there are some viruses that actually have instructions to do the nuclear membrane. And you can see how this works from a nucleus, and same type of thing, but it uses the nuclear membrane, and then it leaves by a modified form of exocytosis. And then after it gets out, the cell membrane falls away, and it just has their envelope. Could use the nuclear membrane, could also use the endoplasmic reticulum membrane as well from that. All right, enveloped viruses must leave by budding. What about naked viruses? Non-enveloped or naked viruses, they don't need a membrane. They typically leave by lysis. they will actually rupture the cell. Remember all of the time during synthesis, the cell cannot maintain itself. It's busy making viral nucleic acid and viral proteins. And so eventually the cell weakens. The virus actually makes an enzyme that can weaken the cells as well, and eventually the cell can't hold itself together and it will rupture, and all of these virions will be released outside of the cell at that point. All right, so release of virions happens two ways, either by lysis or by budding. All right, let me show you this. So what controls how many virions get made? Could be the size of the virus. Some viruses are very small, and so there is enough material in the cell to make a lot of them because they're very small. The larger viruses, it's going to take more building materials, if you will, so they can't make as many of them. Depends on the cell. If the cell is very healthy and robust, it can make a whole bunch. So the number of virions released can vary some due to that. This pox virus is a pretty large virus, and it typically can make 3,000 to 4,000 virions for every cell it invades. So polioviruses, that's even more scary. Polioviruses are very small, and they can make up to 100,000 virions. So let's look at this chart, because this is kind of scary. Over here, I've got number of infected virions. So you come in contact with one poliovirus. One virion, you pick it up somewhere, gets inside, and you can even think of it as SARS-CoV-2. You come in contact with one. That one virion finds a host cell with a receptor. Absorption happens, penetration happens. Now the number of infected virions in the environment has gone to 0 because it's inside the host cell. So it's not in the environment anymore, it's inside the host cell. Time is elapsing for synthesis and assembly, and again that varies depending on the virus, depending on some other factors. But eventually that host cell is going to be so damaged it can't make any more virions, and release is going to occur. If release is occurred by lysis, immediately you get all of these virions, maybe up to 100,000, being released into the environment. When they are released, then they can be carried by currents, air currents, blood, lymph fluids through the body, and every time they come in contact with chance collision to a cell that has the appropriate receptors, they can start the cycle all over again. So you can see how quickly viruses can proliferate within our bodies and how quickly this can happen, and the virus can take over from that. If it's released by budding instead of by lysis, we call this shedding viruses. Instead of lysis, where everything goes out all at once, this happens slowly. So as virions are made, this process will happen, and then these viruses are shed. So the advantage is the host cell stays together a longer time. It could potentially make more virions during this time period, and if the virions are released slowly, there can be some advantage in that as well, versus all the virions being put right in one geographic location. They can be shed in multiple locations multiple times and may have an advantage in finding a new host that way. Okay, so synthesis and release, though that is the last stage of the viral multiplication. Thanks.